BIO203 Transpiration Lab. Setting up the potometer. Secure the plastic pipette to the lower clamp. Attach the syringe to the plastic pipette. Fill the potometer with water from the tray. Be careful not to suck up any air bubbles. And keep the end of the tube submerged in water. The water level in the pipette should be around the 0 mark. It's a bit higher here, but that's fine. Cutting and inserting the plant stem. Keeping the tip of the stem underwater, use a diagonal cut to remove a small portion of the end of the stem. Keeping the tube and the cut end of the stem underwater, insert the stem into the tube. Be careful not to let any air get into the tube. Carefully dry the outside of the tube and stem. Also dry off any leaves that may have gotten wet. Add a generous layer of Vaseline to create an airtight seal between the tube and the stem. Secure your sealed plant under the light box on the retort stand. Adjust the plant level and position as necessary under the light box. Remove the syringe from the end of the pipette. If the water level in the pipette starts to drop rapidly, or the water in the tube drops below the stem, there is probably a leak in the system and you will have to start again. But if everything looks good, turn on the light box. Record the starting level of water in the pipette and start your timer. In this demonstration, the time 0 reading would be approximately negative 0.13 milliliters. Remember that we started a bit above the 0 mark in this demo. As time goes on, the water level in the pipette should slowly go down. For each time point, record the water level in the pipette. Continue to take pipette readings every minute for the first 5 minutes, and then take readings every 5 minutes for the next hour. In between the 5-minute readings, you can move on to the next part of the lab. Leaf surface peels. Use 2 leaves from the same type of plant that you used to set up the potometer. We're using the C3 bean plant here. Use the abaxial or lower side of one leaf and the adaxial or upper side of the other leaf. Use clear nail polish to paint 3 small patches on each of the upper and lower leaf surfaces. While waiting for the polish to dry, label your slides with the plant type and leaf side. When the polish is dry, use a small piece of clear tape to carefully remove each patch of nail polish. and attach them to your labeled slides. You can now use your microscope to examine the surface peels and do your stomator counts. Leaf surface area. After taking your last water level reading from the potometer, remove your plant and bring it to one of the photo stations. Cut off all of the leaves. And arrange them on the pink paper with a ruler and label. Cover with a sheet of plexiglass to flatten everything out. And take a picture using your phone. Transfer the picture file to the lab computer and use the ImageJ software to determine total leaf surface area.